Here we will discuss about the process, different types of transpiration and different experiments which are related to this and the factors which are affecting transpiration. Now, what is transpiration? Transpiration is the process in which water in the form of water vapor is lost from the surface of the aerial parts of the plants, mainly the leaves. So, here in this, water is lost from the aerial parts of the plant in the form of water vapor, and these are generally lost from the leaves. That is what we call, or it is also known as transpiration. Now, let us see some experiments which shows that transpiration takes place from the leaves. Here we have some spinach. Spinach leaves are taken. Spinach leaves are taken. So, freshly prepared, these are fresh spinach leaves. Before we start the experiment, we will wipe the leaves with blotting paper so that no water is sticking to the leaves. Then we will put the leaves in a polythene bag and we will tie the bag properly so that no air can enter into the bag and leave it there for few hours. And what we will see after a few hours, we will find that in the inner portion of the polythene bag, we will find some droplets of water getting collected. From where these droplets of water has come inside the bag? The leaves were wiped dry. So, surely it has come from the tissues of the leaves. The tissues in the leaves, they lose water in the form of water vapor which comes out from the leaf surface and gets condensed in the inner layers of the polythene bag. That shows that the leaves are losing water in the form of water vapor. Let us say another experiment. Here we take a potted plant and this plant is covered by a polythene bag. It is a well watered plant. It is covered with a polythene bag. See here, this is covered with a polythene bag, and then is another pot where there is no plant. And these two setup are kept outside in the sun for some time. Then what we observe? We observe that in the inner layers of the polythene bag, there are water droplets getting collected. So, from where these water droplets have come? These water droplets has come from the leaves. The leaves lose the water in the form of water vapor. This comes out in the form of water vapor and gets condensed in the inner layers of the polythene bag. As they accumulate or they get condensed, so we find them as water droplet. This shows that the plant loses water in the form of water vapor and it accumulates in the inner parts of the polythene bag. But here in this, the second one where there is no plant, no such accumulation is seen. Let's go to the next part. Is another experiment using a bell jar. This also demonstrates transpiration. Here we have 3 bell jars. In the first one, we have a potted plant. Is a well watered potted plant, and the lower portion of the pot, that is where the soil is covered with a polythene bag. In the second one also the same type of setup is there, but inside we have a wooden stick. On the wooden stick we have this cobalt chloride paper. We have the cobalt chloride paper kept inside it. In the third one, there is no plant, only the wooden stick and the cobalt chloride paper. Now let us see what result we will get. Now in this first setup, what we find after a few hours, because this 3 setup is kept in the sun for a few hours. In the first, there is no cobalt chloride paper, but we find in the inner portion of the bell jar, there are droplets of water getting collected. In the second one also, there is droplets of water being collected. But the cobalt chloride paper which is there inside the second one, this cobalt chloride paper turns to pink. It turns to pink, confirming that the droplets of water which has been collected is nothing but water. And in the third one, there is no plant, so the cobalt chloride paper which is there, this remains blue in color. There is no change in the color of the cobalt chloride paper. So that shows that there is no condensation of water. So the water is not present inside the bell jar. This is used as a control to verify the other two, that the water which has been accumulated in the first 2 setup did not come from the bell jar. So this experiments proves that plants give out water in the form of water vapor from the aerial part, that is the leaves, and that is what we call as transpiration. Next, go to the next part. How do we measure transpiration? There are 2 ways of measuring transpiration. One is the weighing method and other is by using a potometer. Let us see one by one. Now in the weighing method, here we take a glass bottle with a side tube. This side tube is graduated. This side tube is a graduated one. So there is a scale here. So as it is filled with water, we can mark the level, we can take the reading, the initial reading, and there is a twig with roots fixed inside it. It is made airtight so that nothing can come out. Before we start the experiment, the whole setup is measured. Weight is taken with the help of a weight. We record the weight, and after the experiment also we will again take the weight. Say beginning of the experiment, the weight is say x gram. This is the initial weight starting, and after the experiment, say after we keep it in the sunshine for few hours, we again record the weight of the experiment, the whole setup, say that is y gram. Now what will we observe? We will observe there is a decrease in the weight of the whole setup. What is the decrease? The decrease is x minus y. This much, this x minus y gram. This is the decrease in the weight of the setup. And here in the graduated stem also we will find that the level of water has come down. That shows what? That the twig has absorbed the water and the water is lost from the leaves by transpiration. Water has been lost. So as the water has been lost by transpiration, there is a decrease in the level of the graduated stem. If the initial reading of this one is say p centimeter cube, after the experiment it will become q centimeter cube. This difference will give the volume of water. How much is the volume of water? Now this volume, when you multiply with the density of water, you will get the weight of the water lost. It is the weight of the water lost. This difference into the density of water. What we will find? That the weight of the volume of the water which is lost is equal to the loss in the weight of the whole setup. That shows that the amount of water which has been absorbed and the loss in the weight of the water is equal. So by this we can, if you record the time, then we can now measure the rate of transpiration. So there is one way of measuring the transpiration. Another is using the Gannon's potometer. Potometer is generally, it is an instrument which is used for measuring the rate of transpiration. It measures the rate of transpiration. It measures the rate of transpiration. So here we have the Gannon's apparatus. Here we have a freshly cut twig with broad lips inserted into the Gannon's potometer. The potometer is completely filled with water, so that no air spaces are left. At the end there is a beaker containing water. A beaker is there which contains water. The end of the Gannon's potometer is kept in the water in the beaker. And with the help of an air dropper, we will insert an air bubble. The air bubble is at the initial level. It is the initial level of the air bubble. And there is a scale. So if the water is absorbed by the twig, as the water will enter, the bubble will move forward. So by measuring the reading here on the scale, we will know how much water has been absorbed by the twig. So the difference in the initial and the final reading, which is the final reading here, this difference will give us the amount of water which is being absorbed by the twig. So after arranging this one, we will put this setup in the sunshine, and after few hours we will take the reading. As we said, there will be a decrease in the level, means the bubble from here, from the initial level, will go to the final level. This difference in the reading will give the volume of water absorbed by the twig. So that much amount of water has been absorbed, and that much amount of water has been lost from the twig by transpiration. So this amount divided by the time taken will give us the rate of transpiration. So by this method, we can record the rate of transpiration. So these are the two ways by which we can record the rate of transpiration. One is the weighing method and another is the potometer method. I hope you understand. If you have anything, you can ask. Now let's go to the next portion of the chapter. As we are going to discuss the different types of transpiration, let us see the structures through which transpiration takes place. One of them is the stomata. What is stomata? Stomata are microscopic pores. These are microscopic pores. These are microscopic pores which are present in the epidermis of the leaves and the young green stems. Generally, these microscopic pores are surrounded by kidney-shaped or dumbbell-shaped guard cells. The guard cells are surrounding the microscopic pores, which are known as the stomata. So here, these are the guard cells. These are the guard cells. These are surrounding here. This is kidney shaped, and the pore here is the stomata. Singular is stoma. If there are plural, then it is known as stomata. It is plural form is stomata. So the guard cells are surrounding the pores. So guard cells are regulating the opening and closing of stomata. We will see in the next part of the chapter. Now these guard cells which are present in the epidermis of the leaves, they have the chloroplast. As we know, chloroplasts are necessary for trapping the solar energy. It contains the green pigment chlorophyll. It traps the solar energy. And these kidney-shaped guard cells are found in the dicot plants. Dicotyledonous plants, they have kidney-shaped guard cells, whereas in case of the monocot plants or monocotyledonous plants, the shape of the guard cell is dumbbell-shaped. So by seeing the shape of the guard cells, we can know whether the plant is a dicot or a monocot. And if you see the guard cells, the inner walls of the guard cells, these are the inner walls. These are thick. These inner walls are thick. Whereas the outer walls, these are thin. So when the guard cells become turgid, they become concave in shape because the inner walls cannot stretch as they are thick. Only the outer walls stretch when the guard cells become thick. So that makes the guard cells to be concave in nature. Next, another structure through which transpiration takes place is the cuticle. Cuticle is the waxy layer on the upper epidermis of the leaves to prevent the loss of water by evaporation. Where do we find the cuticle? On the surface of the leaves. You will see on the surface of the leaf there is a waxy layer, shining layer is there. That is the cuticle. This cuticle is this cuticle is generally made of a waxy layer. A waxy cutin is there, is a waxy substance called cutin is present in it, and this cuticle is sometimes thick and sometimes thin. Though the cuticle is mainly present to prevent evaporation of water from the leaf surface, but still some amount of water gets lost from the surface of the leaves. So as it gets lost from the surface of the leaf through the cuticle, it is called cuticular transpiration. So cuticle is also the structure through which transpiration takes place. Another structure is the lenticel. Now the lenticels are aerating pores. Lenticels are aerating pores. The lenticels are aerating pores. Aerating pores means pores through which air can pass. Pores through which air passes. So they are generally found in the woody stem. They are found in the woody stem, and these are actually loose mass of parenchyma cells. So gaps are created that creates the lenticels. So lenticels generally help in respiration because that also helps in the exchange of oxygen and carbon dioxide between the air and the plant. Besides this, as they are always open, they also allow some loss of water in the form of water vapor. So this kind of transpiration which takes place through the lenticels are known as lenticular transpiration. And lenticels never closes, they remain open throughout the day and night. Now let us see the different types of transpiration based on these structures which we have studied. Three types of transpiration are there: stomatal transpiration, lenticular transpiration, and cuticular transpiration. So three types of transpiration we can see taking place in the plants. Let us see them one by one. Now, stomatal transpiration. What is stomatal transpiration? It is the loss of water in the form of water vapor from the leaves through the stomata. That is what we call stomatal transpiration. So, water is lost from the leaves through the stomata in the form of water vapor. That is what we know as stomatal transpiration. Now, 90% of the water which the plants lose by transpiration actually takes place by stomatal transpiration. So, maximum water which loses from the plant is through the stomata, and it generally takes place during the daytime because stomata remains open during the daytime. Stomata is meant for taking in carbon dioxide from the atmosphere because carbon dioxide is one of the raw material for photosynthesis. So, stomata generally opens during the daytime to take in carbon dioxide and give out oxygen and help in the process of photosynthesis. But along with that, as it remains open, water is also lost from the plants through the stomata. Actually, maximum of the water which gets lost from the plants takes place through the stomata. That is what we call stomatal transpiration. Now, the second part of the stomatal transpiration. Water is generally absorbed by the roots of the plant, and this water rises up through the stem and reaches the leaves. Through the stem, it goes through the xylem and reaches the leaves where the veins take it to the leaves. So, water reaches the leaves, and in the leaves, what we find? We find there is spongy mesophyll cells, the inner tissue between the upper epidermis and the lower epidermis. Between the upper and the lower epidermis, that portion is known as the mesophyll tissue. There are some loose mass of parenchyma cells which makes up the spongy parenchyma. So, this loose mass of cells, these are the spongy parenchyma cells, or we call it spongy mesophyll tissue. So, there are many intercellular spaces. Many intercellular spaces are there between the spongy mesophyll cells, and the cells which are there in the mesophyll tissue, they give out the water in the form of a thin film. It comes out and sticks to the surface of the cells in the form of a thin film, and this thin film of water evaporates and fills up the intercellular space in the form of water vapor. So, it gets saturated. As the water evaporates, the gaps, the spaces which are there between the cells, they get filled up with this water vapor. Then this water vapor diffuses to the next area where it is available, the connecting intercellular space, because we know water moves from a region of higher concentration to a region of lower concentration. So, the water vapor molecules, they move from a region of saturated region to a region where there is less. So, all the intercellular spaces which are available, the water moves and it escapes, ultimately reaches the stomata and escapes into the air. This is how the stomatal transpiration takes place. This is the pathway. From the roots, the water is absorbed, reaches the leaves, and from the leaves it is lost through the stomata, it goes out. That is how the transpiration, stomatal transpiration takes place. Now, let us see how the stomata opens and closes. There are two theories which describes the opening and closing of stomata. One is the potassium ion concentration theory, and another one is the sugar concentration theory. So, we will see them one by one. First here it is the potassium ion concentration theory. During the daytime, the chloroplasts present in the guard cells perform photosynthesis and leads to the production of ATP. So, as we already said, there are chloroplasts present in the guard cells. So, as the sunlight falls during the daytime, this chloroplast, they trap the solar energy and perform photosynthesis. They produce the glucose, and the glucose is utilized to generate or produce ATP. And this ATP is utilized to pump in potassium ions from the adjacent cells. The adjacent cells, from the adjacent cells, potassium ions are pumped into the guard cells. As the potassium ions enter the guard cells, the concentration of the cell sap becomes concentrated and it becomes hypertonic. So, the guard cells become hypertonic because of the entry of the potassium ions. More and more potassium ions are entering, utilizing the ATP, and that is making the guard cells to become hypertonic. When the guard cells become hypertonic, water is drawn from the adjacent cells. Water from the adjacent cells now enter into the guard cells. From the surrounding cells, they enter into the guard cells. As the water enters into the guard cells become turgid. So, as the guard cells become turgid, what happens? The guard cells expand or swell up, and so the inner walls which are there, they move away from each other and the stomata opens. The gap between the walls is created and the stomata opens. So, that is how the stomata opens during the daytime. And at night, when the sun goes down, there is no more light. Light is not present. In the absence of light, no photosynthesis will be able to take place. As no photosynthesis can take place, no ATP is produced. So, as there is no ATP, what happens? The potassium ions cannot be held in these guard cells anymore. Already the concentration of the potassium ions is more compared to the neighboring cells because they have been pumped in from the neighboring cells using the ATP. Now, there is no ATP to hold them back. So, all the potassium ions now leak out. As they move out from the guard cells concentration, the cell sap concentration of the guard cells decreases, and now there is more water and less ions in the cell sap. So, the guard cells become hypotonic. Hypotonic means they have now more water compared to the neighboring cells. So, the guard cells now lose water by exosmosis. Water goes out into the neighboring cells. The guard cells now become flaccid. As the guard cells become flaccid, they cannot remain in that curved position anymore. They become very weak and limp. So, the inner walls now come closer to each other and this stomata gets closed. So, stomata closes as the inner walls of the guard cells come closer to each other. That is how stomata closes during the night when there is no sunlight. Let me repeat once more, this is an important theory. So, first is again I am telling that during the daytime, the chloroplasts which are present in the guard cells, they perform photosynthesis, they produce the glucose. As they produce the glucose, this glucose is utilized to produce ATP. The ATP is then utilized to pump in potassium ion into the guard cells. So, as the potassium ions enter the guard cells, the cell sap concentration becomes hypertonic. As it becomes hypertonic, water is drawn into the guard cells again. The guard cells become turgid, it becomes concave in shape, and so the stomata opens. At night, when there is no sunlight, no photosynthesis, so there is no production of ATP. As there is no production of ATP, the guard cells cannot hold the potassium ions anymore. So the potassium ions leaks out. As the potassium ion leaks out from the guard cells, the concentration of the potassium ion decreases. The guard cells now become hypotonic. So water from the guard cells are lost by exosmosis and the guard cells become flaccid. As a result, the stomata closes. So this is the potassium ion concentration theory. I hope you understand. Next we come to the sugar concentration theory. So in the sugar concentration theory, the same thing, guard cells, they have chloroplast in them. As they have chloroplast, the guard cells perform photosynthesis that leads to the production of glucose. That leads to the production of glucose. It is similar, but here the glucose dissolves in the cell sap, and because of the glucose dissolving in the cell sap, the guard cells become hypertonic. And as the guard cells become hypertonic, water enters into the guard cell by endosmosis, and the guard cells now become turgid. As they become turgid, the stomata pore opens. So that is how stomata opens during the daytime. Again, the guard cells contain chloroplast. They trap the solar energy, performs photosynthesis and produces glucose. Glucose dissolves in the cell sap and the guard cells become hypertonic. As it becomes hypertonic, water enters into the guard cell from the adjacent cells by endosmosis. The guard cells become turgid, the inner walls move apart and the stomata opens. At night, when there is no sunlight, no photosynthesis can take place. As no photosynthesis takes place, the concentration of carbon dioxide increases because respiration is continuously taking place in the cells of the leaves, and that is increasing the carbon dioxide. During respiration, the carbon dioxide which is produced is also utilized during the photosynthesis process. But at night, as there is no photosynthesis taking place, that the carbon dioxide which is produced in the guard cells cannot be utilized, and as a result, this carbon dioxide concentration increases. It dissolves in the cell sap and it leads to the production of this carbonic acid. Carbon dioxide dissolves in water and forms carbonic acid. Carbonic acid now converts the glucose into starch. The carbon dioxide which is there, it dissolves in the cell sap of the guard cells, converts into carbonic acid. The carbonic acid converts the glucose into starch. And we know starch is not soluble, it's insoluble in the water. So starch being insoluble, the cell sap now becomes hypotonic because glucose was dissolved in the cell sap, that made it hypertonic. All the glucose has gone out, so the cell sap has now become hypotonic. That means the percentage of water is more now. So water from the guard cells will now move out. As the water moves out from the guard cells now become flaccid. And as the guard cells become flaccid, the stomatal pore closes. That is how stomata closes during the night. So again the closing part, at night no light, no photosynthesis, so there is an increase in the carbon dioxide concentration. The carbon dioxide concentration increases means it dissolves and forms carbonic acid. The carbonic acid converts the glucose into starch. The starch is insoluble, so the cell sap become hypotonic. And as the cells have become hypotonic, water is lost by exosmosis. Guard cells become flaccid and the stomatal pore closes. I hope you understood the opening and closing of stomata. Next we come to the lenticular transpiration. As we already discussed about lenticels. So the loss of water from the lenticels in the form of water vapor from the woody stem is known as lenticular transpiration. And as the lenticels remain open throughout the day and night, so continuous loss of water takes place. But the loss of water which takes place through the lenticels is very much less, only 1 to 2% of the total transpiration taking place in the plants. So it is very much minimum, but some amount of water is lost through this. Next, we have the cuticular transpiration. As we said, cuticles are present on the surface of the leaves to prevent the loss of water, because this waxy layer prevents water loss. The leaves generally are exposed to the sunlight to get maximum sunlight. So this cuticle which is present on the surface, they are meant to prevent the water loss, but still some amount of water gets lost from the surface of the cuticle. That is what we call the cuticular transpiration. So water vapor comes out from the surfaces of the leaves. So some amount of water is lost from the plant by this process, and cuticle generally serves to prevent evaporation of water, but still some amount of water loss takes place. That is why we call it cuticular transpiration, and it depends upon the thickness of the cuticle. Greater the thickness of the cuticle, like in case of banyan tree, we see banyan tree leaves have very thick cuticle, and that will prevent maximum loss of water. So there will be lesser transpiration. More thicker cuticle, lesser is the rate of transpiration. If there is a thin cuticle, higher will be the rate of transpiration. So we have seen the three types of transpiration. Now let us see the factors which affect the rate of transpiration, or how is transpiration affected. Now there are certain factors. There are two types of factors which we see. One is the internal factor and other is the external factors. Internal factors and external factors. This affects the transpiration rate. The first internal factor is, we are going to discuss initially the internal factor. The internal factors, let us see one by one. Leaf structure, the structure of the leaves. What are the anatomical features of the leaves? Now there are different types. In some leaves, the stomata remains sunken. In some leaves, the surface of the leaves is covered with epidermal hairs. In some leaves, there is a waxy layer of cuticle. In some leaves it is thick, in some leaves it is thin. Then the mesophyll tissue which is there, in some leaves it is compact. Air gaps are very less, no intercellular space. So if these are there, that will reduce the rate of transpiration. In some leaves there are less number of stomata. So if they are present, in such cases, the rate of transpiration will be affected. It will reduce the rate of transpiration. So if the leaves have sunken stomata, they will have lesser rate of transpiration than normal stomata. If the surface of the leaf is covered with epidermal hairs, they will have a lesser rate of transpiration compared to the leaves which are not covered with any hair. If the surface of the leaf has a very thick cuticle, they will also perform lesser transpiration compared to the leaves which have a thin cuticle. Then in case of compact mesophyll, if there is no intercellular space between the mesophyll tissue, then there will be a lesser rate of transpiration. If the number of stomata is less, that also will reduce the rate of transpiration. So the anatomical structure of the leaf plays an important role in deciding whether transpiration will be higher or lesser. Second factor is leaf area. Some leaves are very broad and some leaves are thin. If the leaves are broad, then transpiration will be faster. If leaves are thin, then transpiration will be minimum. Third factor is water content of the leaves. If the leaves do not have sufficient water, then suppose the water which is being absorbed by the roots, the plants are not being able to absorb sufficient amount of water which the leaves need. So the leaves will have insufficient water. In such cases, the leaves will now undergo wilting. The cells of the leaves will become flaccid and it will undergo wilting. In such cases, when the leaves wilt or the plant wilts, especially we can see in case of smaller plants during the noon time, you see them, the leaves become very soft and they limp. In such cases, the rate of transpiration will get reduced. So if there is lesser content of water, there is a mechanism in the plants which will help the plants to reduce the rate of transpiration and thereby help the plants to survive such conditions. Fourth factor, orientation of the leaves. How are the leaves arranged? If the leaves are arranged transversely, that means if the flat surface is facing the sun, in that case, if the leaves are facing directly, the flat surface is facing the sun is there. If it is a flat surface, so more rate of transpiration will be there. And so as there will be a higher rate of transpiration because surface area is more, so more transpiration can take place. But if the leaves, instead of being transverse, if they are perpendicular, the sun is here and they are perpendicular, they are not facing the sun. In such cases, the rate of transpiration will be slower. They transpire at slower rate if they are placed perpendicularly. So how the leaves are arranged in the shoot, that also determines whether the rate of transpiration will be faster or slower. Another point is succulence. Presence of some mucilage or latex. Some plants we see there is some sticky substance present. If you break the leaves, you find that sticky substance is present in them, or you'll see milky latex, which you see in case of euphorbia or rubber plant, whatever we call it. So in such plants, the rate of transpiration will be less. It will reduce the rate of transpiration compared to the normal plants. External factors. The external factors which affect the rate of transpiration are light, temperature, humidity, wind, velocity, carbon dioxide, soil condition and atmospheric pressure. Let's see one by one. Light intensity. When we say light intensity, if there is higher intensity of light, stomata will remain wide open, so that will increase the rate of transpiration. Second factor, temperature. If the temperature is higher the temperature, faster is the rate of transpiration. Third factor, humidity. If there is a higher humidity, means more moisture in the air, lesser will be the rate of transpiration because the air is already saturated with moisture. So the air will not have a capacity to hold or take in more humidity or more moisture into the air. So rate will decrease. There will be a lesser rate. High humidity, lesser rate. If the air is dry, then higher will be the rate of transpiration. Wind velocity. If the wind is still or air is still, then rate of transpiration will be less. If the humid air is replaced by dry air, then as the wind will blow more, so rate of transpiration will become faster. So when there is a wind blowing, generally we find that rate of transpiration is higher. The other factor is carbon dioxide. Generally, normally we have 0.03% of carbon dioxide in the air. But if the level of carbon dioxide is more, then stomata generally closes, and that will result in the decrease in transpiration. Sixth factor is the soil condition. If the soil has plenty of water and the roots will be able to absorb more water, in such cases there will be a higher rate of transpiration. Greater would be the transpiration if the roots are able to absorb more water. And third, last factor, the atmospheric pressure. The rate of transpiration is inversely proportional to the atmospheric pressure. If the atmospheric pressure is high, if it is more, then the rate of transpiration will be less. So we have discussed certain portion of the chapter. There are some more parts left, as we already taken a long time. So I am not continuing more. If you have any doubt, you can see the video again. We will try to clarify it in the later part. So thank you.